Hello everyone, today I'm going to talk a paper called Intense Threat Switches Dorsal raphe serotonin neurons to a paradoxical operational mode. So this paper proved that our brain operates in a fundamentally different way under intense threat. Seal et al. Found that the mice paused when serotonin neurons were transiently stimulated in low or medium threat environments, but this same neural population was stimulated in high threat environments. Mice tried to escape. So basically they found there's a group of neurons operate differently in different environmental conditions. So under high threat, under high threat and low or no threat, this neuron fire differently. And they tested two groups of neurons, one called dorsal raphe nuclei, serotonin, and another is dorsal raphe nuclei, GABA. Let's look at the abstract. Survival depends on the selection of behaviors adaptive for the current environment. And the mouse should run from a rapidly looming hawk, but should freeze if the hawk is coasting across the sky. Previous study shows that serotonin has been implicated in adaptive behavior. Environmental regulation of its functional role remains poorly understood. And they found that in mice, the stimulation of this group of neurons suppressed the movement in low and moderate threat environments, but induced escape behavior in high threat environments. And this movement-related dorsal raphe serotonin neural dynamics inverted in high threat environments. Stimulation of dorsal raphe GABA neurons promoted movement in negative but not positive environments. And this movement-related GABA neural dynamics inverted between these two different environmental conditions. So, these dorsal raphe circuits switch between two distinct operational modes to promote environment-specific adaptive behaviors. To summarize the results of this paper, I made this table. Under no or low threat environment condition, if you stimulate dorsal raphe serotonin neuron, it will suppress the movement of the mouse. But under the high threat environmental condition, if you stimulate the same group of neurons, it will promote the movement. And if you record in the activation state of this neuron, and under no or low threat environmental condition, it is negatively correlate with the movement and positively correlate with the movement under high threat. And they use the same experimental set, but this time they tested the dorsal raphe GABA neuron. Under negative environment, if you stimulate this dorsal raphe GABA neuron, it will promote movement. And under positive environment, it has no effect on the movement. Under negative environment, the activation of GABA neuron positively correlate with the movement, and it is negatively correlate with the movement under positive environment.